Good afternoon, everyone. Good evening from London. I'm Claudia's brother. I'll be chairing today the talk. We have with us today Juliana Gonzalez-Tobon. She is a Ph.D. candidate of plant pathology and plant microbiology at Cornell University, where she studies bacteria that infect potatoes and how they sense the environment. She's originally from Colombia, where she earned 2 bachelor's degrees in biology and microbiology, respectively, and a master's in biological sciences at Universidad de los Andes in Bogota, Colombia. Aside from her research, Juliana is a writing presenting tutor at Cornell, where she helps other multilingual graduate students with their English and has created a community in social media to communicate science to the general public, mainly in Spanish, and to support each other with tips and challenges about academia. So Juliana, whenever you're ready, take it away.
Sounds good. Thank you, Jose. Well, hello, everyone. I'm happy to be here. We have been trying to schedule this talk with Claudia for a while, so it's finally here. Today, I'm happy to talk to you about potatoes and basically what makes a potato sick. You'll see it's impossible for me to cover everything, but fortunately, I've had experience both in my work back in Colombia and now in my work here at Cornell with this crop and why it's so important. So I hope you enjoy it, and I hope you can learn something new today. Okay, so to start, this talk will be a little bit of a mixture between stories and also data. So if you look at this picture, you probably wonder, what is it about? Or maybe some of you that already know the story can tell me. But this picture is actually very related to these memorial statues that you can find in Boston, Massachusetts. And they all are there because of the Irish potato famine that occurred in Ireland in 1845 to 1852, which was, a very humid summer that made it the perfect environment for a specific pathogen that we're going to talk about later in this talk to destroy the potato crops that these people relied on for their just life. And that's why about 2,000 people, I think, or more, migrated from Ireland to the United States. And one of the ports where they got here was Boston. And that's why this is one of the cities that has a memorial. And you can see the two sides of the story, the families that stayed there or sadly perished during the famine, and then the families that came here or moved to other places to sort of look for other opportunities. So the pathogen that actually was responsible for this is still important nowadays. There are pieces from New York Times articles from 2019. They actually say fungus, but I hope that by the end of this talk, you'll know why this is a mistake. But they basically were talking about something very, very destructive that was attacking tomato plants. And then this is an internal communication here at Cornell, where one of the researchers says, he was around the Ithaca area, and they noticed that the plants that were being sold in the grocery stores were very sick, and people were still buying them, and using them to cultivate tomatoes in their own backyards, which is really very common here in this town. So you'll say, why? But you're talking about potatoes. We're now jumping to tomatoes. I don't want to confuse anybody, but I bring this story up because this is exactly the same pathogen that back then in 1845 made such a huge mess in the potato, industry or world, and now 2009 was attacking tomatoes, basically in all the northeast of the United States. And I'm gonna go back to this later. Then other things that have started to happen recently are that potato growers also here in the northeast of the U.S. were finding their potatoes to look like this in the moment of harvest, and that turned out to be actually because of a different pathogen that's called Dickia, and I'm gonna also talk about this more a little bit later. But it's not considered one of the most recent and also most potentially dangerous threats to this industry in the U.S. It's found everywhere in the world, and it's also rising in the sense that it's beginning to be identified in other places as well. And nobody would like to buy or even consider eating a potato that looks like this. So why do I bring these stories today? Because I think for some of you that maybe are not familiar with the world of plant pathogens, plant health, just general how plant health relates to our own health, you might be wondering, why is it important and why we study plant diseases? Well, I already showed you some of these examples, but there's other reasons why we do this. And people like me that are plant pathologists, usually when I try to explain this to a general audience, or when I'm doing outreach activities with kids, I say I'm sort of like a plant doctor, because we take care and we try to understand the symptoms that plants are showing or crops are showing and why we need to solve them, right? Why we need to control them. So when is the plant considered sick or diseased? Well, the basic definition is just when it's not functioning normally, but that can imply different things. It can imply things like this one here, that the leaves are turning necrotic and are dying. So basically they're not going to be as efficient, as good for photosynthesis as they are when they're healthy. But you can also see, as I showed before, the tubers or the fruits just rotting from the inside and being completely unviable for feeding us and for just commercializing them. And this can be biotic, as I've been mentioning, pathogens, right, that we're going to go more in detail in a minute, or abiotic, so like too much sun, too much water, the use of fertilizers in inappropriate ways, or just too much concentration of chemicals that you're using. And again, why would this be important? Well, you already saw examples of epidemics that have been devastating in their specific places where they occurred and had impacts elsewhere, but also because diseases are everywhere. And I think after this pandemic, people have been more and more familiarized with the fact that pathogens exist, they can make us sick, and we're trying to make it evident that it can also make our food sick. And that's one of the things that we have to really worry about. This is something that we call the disease triangle, which is very important to understand plant diseases, because for a disease to cure, we need it to be, you need to have the pathogen, and for it to be able to attack, we need to have a host plant, and for it to be susceptible to that attack. And then we need that the environment is conducive to that. And maybe you remember that I mentioned in the first example, the summer of that year was very humid. And that was sort of the trigger that made it the perfect place for this pathogen to thrive. Then there's a fourth component that we have, people have included more recently, that it's time. As you have also probably gathered from this pandemic, it's different if you catch a disease early on, or if you catch it, when your whole crop is already showing symptoms. And for a person that their whole life and their economic sustenance depends on that, well, that will be a huge difference. So in my case, I'm a plant pathology that studied this in a molecular level. So I work with cells and all those very small things, but very important, that in this case can be the determinants of whether an infection is successful for the pathogen, meaning that our plants get sick, or it's not successful for the pathogen. Maybe the plant finds a way of how to defend itself, and then the process does not go as well. Here you can see some spores of a fungus going inside a plant, between the plant cells. And just exactly the level of detail where my work usually focuses on to understand these different things that I just mentioned. And then this has been combined with a more recent, explosion of technologies that we can use. Now it's much more cheaper to, for example, sequence a genome and understand all the instructions that each one, the pathogen or the plant, has that are important for this situation to occur or not occur. So taking all of this account when later on, when I talk a little bit more about my research itself. So let's just bring potatoes for a minute, because they're our main protagonist in this talk. This crop, was originated and domesticated in the Andes in South America. It's the third major crop in the world, only after rice and wheat. So you can imagine how many people in the world consume potatoes. It's actually 2% of the calorie intake of the whole population. So that's a huge thing. In the world, more than 300 million metric tons of potatoes are cultivated per year. That's a lot. And part of the reason for this is that it's very easy to cultivate. Because you can propagate it vegetatively, so you don't need like sexual interaction to have it. You can just have sort of clones easily cultivated. That's also why it's cultivated in more than 100 countries. But this also brings sort of a trade-off or something that we have to take into account when that happens. And I'm going to mention it in a minute, which is it gets really very sick. And part of the reason you can be imagining is because if you're propagating it vegetatively, then you will mostly not have genetic diversity. You will have different organisms or different plants that are very similar to each other. So when a threat comes in, if you don't have already the tools to defend yourself from that, well, the plant doesn't have the tools to defend itself from that threat, then you can have a whole crop lost very rapidly. And you'll see how quickly in a minute. And also, all the different types of pathogens can attack potatoes. And this makes me remember when I was a teacher, like an assistant teacher back in Colombia, helping out with the lab of plant pathology. One of our quiz questions was, for the students to pick a crop they consider to be really unlucky in the sense that it gets really sick. And most of them chose potatoes. And I think it's because they saw during the semester that there are so many things that can attack them. You can have fungal diseases, you can have oomycetes, which is what we're going to talk about in a minute. Bacteria, also going to talk about this. Viruses, which I assume a lot of you already know a lot more about viruses than you did 2 years ago. And then nematodes, which are like very little small worm-like animals that can literally get inside the potato roots, for example, or the stems, and make a whole mess there. These are just different pictures of examples of different things that can affect them. But we're going to talk about two in a specific, which are the ones that I've worked with during my almost 10 years, a little bit less than 10 years working in this area. The first one is Phytophthora infestans. That's its scientific name, but it's the pathogen responsible of the first two examples that I showed. So the Irish potato famine, as you can see here, and then 2009 outbreak, which meant that it's still a threat and it's still something that we're dealing with everywhere where potatoes are cultivated. So here in the US and exactly back in Colombia, there are small differences of the populations that we have, of course, but it's still a threat in both cases. This name exactly means plant destroyer. So you can imagine why they decided to name it like that. It's able to affect the whole plant because, just a detail here, some pathogens focus on, only the fruits, only the roots, only the leaves. This one can affect absolutely everything. And that means that the crops are lost entirely very quickly. As I was sort of hinting before, the pathogen is actually an oomycete, which might be a word that's not familiar to a lot of people. In practical terms, it looks really very much like a fungus. So that's probably why the New York Times people called it like that. But technically, taxonomically, it's an oomycete. So it's part of a different group when we look at their evolution and where it came from. It has differences in their cell wall and their genetics, but it looks pretty similar to a fungal pathogen. In common terms, sort of, this is the name that the disease gets, late blight. So if you know potato growers, that's usually the term that people are more accustomed to use. This is an example of how the leaves start to look when it's getting distributed along the plant. And then this is a picture, well, 3 pictures took by Bill Fry, which is an eminence in this area, and he studied Phytophthora for a long time. And he took a picture the first day he went to a crop here in Ithaca of potatoes, and you can see, well, maybe not because of Zoom, but later on you can see that there's some of the plants that look different, and they start looking like less green, more yellow. So he just went there, took the picture, and then a week later he went back, and this is how the crop looked if you didn't do anything to control it. And then 1 week later, the exact same crop, this is how it looked. So this is just to show you how fast a destroyer this pathogen is. And because of that, one of the things we really need to do to control it is to use fungicides and other chemicals. And that's where my investigation back in Colombia came to work and to focus on. So to tell you that, I'm just going to tell you a quick story. These are actually pictures took from Dr. Bill Fry's lab that I was just mentioning him here at Cornell, when they were studying how this pathogen was sensitive to different fungicides. They wanted to know, well, if this is our way to control it, then which fungicides do I have to use and in which concentrations? So this is, for example, one of the lineages that they were using. They were growing it in a petri dish with media, but without any fungicide, so just normal growth. And then this one here had 5 micrograms per milliliter, which was like a semi-lethal dose. So it was harmful, but not super high. And you can see how the growth is much more restricted, but there is growth still. And then this was considered the lethal dose. We're using no growth. Well, something happened, and the person that was making the transfers to repeat the experiment, instead of gathering, the mycelium from a plate that has never been exposed to fungicide, they gathered it from the 5 one, so the middle one. And then when they made the transfers to the different concentrations again, they saw that now it was able to grow as if there was no fungicide, even in the one that was very toxic for it and that was not supposed to grow there. So this generated a lot of questions. They started doing research to understand if this mistake was just a mistake or if it was like a scientific phenomenon behind it. And they figured that it was, and I'm going to talk about it more in a minute, but it's very similar to what happens with antibiotic resistance. It's not the same, but you might know that when you get sick and the doctor prescribes antibiotics for you, they usually tell you, please, take the whole bottle or finish up all the doses, even if you're feeling better, because that's the way to really finish with the bacterial population that's making you sick and not leave some of them that might have characteristics that make them resistant to that antibiotic, so that the next time that you get sick, that antibiotic is no longer useful for you. So it was very similar. We knew that Phytophthora had this natural resistance, but we didn't know that you could sort of trigger it like this. And that led to 2 different papers that are out there if you want to read the data more in detail, because I honestly don't have time to get into it. But we realized that it was only happening with a specific fungicide, which unfortunately is very much used here in the US and also back in Colombia. So that's bad news. It was also occurring in different species of these pathogens, so not only infestants, which is the most popular. And there, it came a moment where I had to finish my part in this research and move here to the US to continue working in potatoes, but in a different disease. So there's some ongoing research from back home, still trying to understand more like, okay, we know this happens in the laboratory, but how bad is it in outside in the field? Does this mean that if growers do not use a fully toxic dose since the beginning, are we gonna promote resistance in our fields, and how much? Then the other thing that is part of one of those papers is that, as I said, I was very interested in the molecular part of it, understanding how the cell reacts. So, it's just visually it's being able to grow, but what is happening inside of it so that it's becoming this resistance. And again, I'm not going into the details of the methods, but after we made a lot of testing and a lot of different things, I got to forming conclusions or things that might be happening here. Part of it is related to the cell, the cells of this pathogen expulsing the toxic substance, in this case, the fungicide, in different ways. And they had all, genes and proteins related to do that. And they were also generating a cellular stress response. So the pathogen in the cellular level becomes stressed and sends signals to the rest, to different parts of the cell saying, this is a threat, we have to expose this, and this we have to activate things that are allowing us to survive, allowing us to grow. One of the things that happened was it was getting able to grow, but it was growing much more slowly. And maybe, those of you that are familiar with biology, you know that we usually see trade-offs, things that allow us to do something, but take us back from something else. And then we also saw different mechanisms of regulation, expression regulation. So some genes being more or less expressed that were related to this. But again, if you want the details for that, you can go to that paper. I think it's really cool, but I'm biased to see that. And then the second thing that happened in my life related to potatoes, and it works really good for this talk, is that when I came here to Cornell, the lab that I started working with was also interested in this crop, because again, it's super important. But they were working with a bacterium, which is called Ichia. And I mentioned it as a third example, a rising threat mostly here in the US and also in some places in Europe. This one works totally different. As you saw, the potatoes look very rot in the inside, and that's because this bacteria uses proteins that are called enzymes, and they're able to degrade everything. They really make a whole smelly potato soup. So it's interesting. The technical name in this case of the disease is called blackleg and soft rot. And the thing is that there's no easy chemical solutions with bacteria. With fungal diseases, at least you have fungicides. With bacteria, there's not something like that. You usually recommend farmers to, watch out for the water that they are using, or the sources of their seeds, or the sources of their plants, but it's hard. And in this case, since these bacteria just degrades everything, there's really not a mechanism of resistance from the plant towards the pathogen. This is how my plants look in the lab when I infect them. It gets really dead on the inside, and then the potatoes just rot a lot. So what am I doing right now? And I don't have like the final data for that. For that, I'll have to return later. But I'm trying to understand how this bacteria sort of smells or detects the plant. And why am I interested in that? Because previous research has told us that this specific bacteria leads its way towards the plant and into the plant based on the chemicals that it sends around them, which are usually chemicals that the plant is secreting because it's feeling attacked. And that's something that happens in general with plant pathogen situations. But this bacteria in a specific has a lot more genes that code for proteins that are in charge of sensing those compounds and generating a response that will eventually make it move, so towards or away from the chemical that is being sensed. So I'm looking this at a genomic level and understanding the mechanisms that regulate these genes to be expressed or not, seeing if there's a way for us to control their expression and therefore, in a way, make the bacteria lost so that it doesn't really know where to go and how to enter. Because different to oomycetes and fungal diseases, the bacteria do not have tools to sort of like pierce their way in through the plant. They need to enter through natural openings or wounds that are already there. Phytophthora, on the other hand, the oomycetes, has tools that makes it like pierce through, and that's why it can infect any part of the plant, because wherever it gets, if the conditions are right, it will just get in. But with bacteria, it's not as easy. So that's why we try to understand what are the differences that make this one such a good colonizer, and what are the things we can do to prevent that. So I'm almost out of time for the research part. And I just wanted to leave as take-home messages. As you can expect, potatoes are one of the most important crops in the world, but they're also one of the most susceptible to stuff and to diseases. Fortunately, we have, there's people working on this, and this can be at different levels, at a disease sort of molecular level, how the interactions go, or more general field-like situation. In my case, my research is more molecular based. But if you're interested in this area, there's a lot of people working in different levels, and it's super interesting to see the different ways we're all trying to tackle these diseases. And then, as always, I think it's super important to recognize the people that have been with us during the process of our research. This is my huge lab when I was back in Colombia. I studied at Universidad de los Andes, which is one of the most important universities in Bogota, Colombia's capital city. And these were the people that were around when I was there. And then when I moved here, these are the other, sort of research family that I found here, the lab of Dr. Melanie Filiatrat, who work, as I said, in bacterial pathogens, not only Dickia, but that's the one that I chose to focus on. So it has been a really nice experience to go from, 2 different places, 2 different labs, 2 different pathogens, but the same crop, and understanding how they're all being a threat to that really, really important crop for us. And then I saw, from previous talks and from Claudia's instructions that it's important for these seminar to talk about our Latin heritage, and I think that's awesome. I am from Colombia, as Jose mentioned at the beginning, and maybe when you think about Colombia lately, you'll picture this if you have been following the Encanto boom, and how it has become so popular on TikTok and just social media in general, and even the Oscars. And that's cool because it's actually a movie that takes and represents a lot of different things from Colombia. But Colombia is so diverse that it's very hard to transmit everything in a single delivery way. I am particularly from the center of Colombia, the capital city. This is Bogota. This is actually close to my university. It was around someplace around here. It's a very big city. It's approximately 7.5 million people. And just for comparison, for people, for example, from the US, it's just 1 million less than New York City. So I think that's a cool, fun fact to give, because sometimes people imagine that cities in Latin America are usually smaller, than in other countries in the world. And well, it's not always the case. A lot of people, it's also really interesting because it's almost 9,000 feet above the sea level, which makes it very hard for people that have lived all their lives in a different, closer to the sea. Actually, when we got married with my husband, we invited friends from the US. They had a really hard time breathing up there, just like going up the stairs, because while you get accustomed to the amount of oxygen that you have there, it's hard. It's the capital city of many things. You can read online, and Clara mentioned, it's also good to talk about these places so that people that have never been in Latin America want to go. So you can research online, but you'll find different, gastronomic things, dance, music. It's honestly, there's something for everybody there. It has been in existence for more than 400 years. So there's also a really cool part of the city of, history-wise and understanding all the process of the colonization on the Americas. And then one of the options was to bring a recipe. This is one of my favorite from Colombia. You also, if you saw Encanto, you probably saw it featured there, which is one of the things that Julieta uses to heal people, arepas con queso. And this is a good recipe, honestly, that I've been using since I'm here in the US, because it's super easy. You just grab harina pan or a different type of cornmeal from the grocery store. And if I can find that in Ithaca, I'm sure you can find that elsewhere in the US. And it's a very easy process. If you like cheese and you like corn, this is 100% a go-to for even a snack or just a meal, like a small meal. It's awesome, honestly. And now, well, I think we can take any questions or comments. This is my Cornell email. That's the one I mostly see. But then also I wanted to say, since Jose mentioned it at the beginning in my intro, ever since the pandemic started, I've made a huge effort to bring science to the general public in social media, mostly in Instagram. It started because of the pandemic and the COVID vaccines, but it has also translated into just other things in science and important about academia, built a community that I'm honestly very proud of, with a lot of mostly women from both Latin America and other Spanish-speaking countries, motivated to either become part of science and academia or are already part of it. We just share experiences, talk a lot about the things that are not usually spoken much in other spaces. So if you are seeing this later and are interested, go and check it out. I honestly really love that community, and I think you might do too, if you identified with the things that I just said. And I think this is it. Thank you so much.